experience working with ICRSAT on legume viruses. He has main interest on virus surveillance, diagnosis, control, phytosanitary and seed quality assurance. He has a few number of awards, the Young Scientist, CJR Young Scientist Award, IITA Best Scientist Award. He is a member of steering group of the African Union Inter-African Phytosanitary Council and Executive Committee of the Plant Virus Epidemiology UK and management team of CJR Gene Bank platform. And he has more than 140 research articles, published book chapters in various international journals, and currently supervised more than 10 doctoral students. Today's topic is diagnostics, phytosanitary regulations, and clean seed systems. Dr. Lavakumar.
Thank you. Thank you, Dr. Krishna Reddy. I'm trying to share my screen. Just hold on a minute.
Take the time, no problem.
Do you see my screen? Okay, so you had to pardon my throat. I have a throat infection. I'm recovering from throat infection, so I had to frequently clear my throat. At the outset, I would like to thank organizers, especially Makesh Kumar, for inviting us to give a presentation and a specific topic to deal with this of diagnostics, especially for phytosanitation, clean seed systems. So I'll be providing a brief overview based on our experience of application of diagnostics in Nigeria. We have a fairly large team, interdisciplinary team working on both virus aspect as well as cassava propagation aspect. Much of the introduction of the previous speakers, right from Makesh Kumar to Dr. James, has provided the base of the cassava viruses. So I would not dwell into much of those details, and I will try to go specifically into the point. So as an outline, I have three elements to share with you. The first one, use of diagnostics for clean seed production for domestic and international seed distribution, and the regulations for seed distribution, both domestic and international, and how they influence our activities and preparation of material. And then finally, the efforts that we are putting in place to strengthen the cassava seed systems, domestic cassava seed systems. James already has provided some details about the work in Tanzania, and I briefly talk about the Nigerian case. Why germplasm distribution is important for us? As you know, as the CGR Center, IITA hosts one of the largest gene banks, perhaps the biggest gene bank in Africa, hosting germplasm food staples. And IITA also has a crops. As a headquarters, bulk of the distributions are made from Ibadan Station, which is located in Oyo State in Nigeria. We have in our gene bank conserving more than 34,000 accessions of banana, plantain, cassava, cowpea, maize, yam, and other legumes. As far as cassava is concerned, we have close to 3,184 accessions. 72% of them are established as clean stock, and they are available for distribution. It includes both land races, breeding lines, and also some wild relatives. I would be only focusing on cassava. So any germplasm distribution activities is a risk because germplasm can carry pathogens as well as pests. It is moved from a country to country or within country. If this material is infested with pests, they automatically get moved along with the material. So this is one of the major contributors for transboundary distribution of important pests. And the example that some mosaic outbreak, although source of introduction is not only to Southeast Asia, but it is very likely that it must have been introduced through planting material. Vector distribution, vectors spread for such a long vectors spread is unlikely. So there are a lot of procedures and protocols involved to contain. And as an international center involved in germplasm distribution, it is imperative for us to follow these protocols rigorously. So we already learned about a number of viruses, both in Asia, Africa, and Latin America. Within Africa, that's where bulk of our activities are focused. Our major concentration is on cassava viruses, cassava mosaic, and cassava brown streak. And we already learned from the previous presentation that there are multiple species involved. For example, there are more than 11 species associated with cassava mosaic disease, whereas in case of brown streak, at least 2 dominant species, and there are many more variants have been reported. In terms of the distribution, cassava mosaic within Africa is widely spread, whereas brown streak is mainly localized in South and Southern Africa, East and Southern Africa. The white flies, which are ubiquitous, they are present almost everywhere. And then the major mode of planting material distribution within the continent is the stem cuttings, which is the biggest risk. And the stem cuttings are also used sometimes for official exchange, especially for countries where they have very limited capacity to receive tissue culture-based planting material. When it comes to diagnostics, we basically use diagnostics for monitoring cassava brown streak spread, especially from East and Central Africa into Western side. So we have an active program together with our colleagues in East Africa, led by James Legg. And then we use diagnostic for seed certification, especially for domestic clean seed systems, looking for both bigamoviruses in the nucleus stalk and the brown streak as a primitive management. And the quarantine monitoring when it comes to the international distribution of planting material. So I don't have to get any details. Although there is only 2 viral diseases, the involvement of multiple species and the high variation in the sequences, it provides a big challenge with regards to having a robust diagnostic tool that can reliably detect all species and there won't be any false negatives. So we always focus on developing diagnostic tests in the 2 contexts. Number 1 is to identify known viruses, for example, the species which have been already characterized. For that, we target a specific test. And as we distribute germplasm and also receive material from different countries, we also have a challenge of looking for material, looking for pathogens that have not been previously characterized so that we don't unintentionally release any new pest because of not thorough testing or due to lack of knowledge. So we also use broad sweep diagnostics to look for both identified as well as, or sometimes even new strain sequence variants. So there are a range of assays used here. Depending on the crop, we use bioassays. We also use serological assays using polyvalent antibodies or monoclonal antibodies in case specific tests. And most often we use nucleic acid based assays, especially PCR for specific tests, and also use PCR for generic tests using generic primers, either level specificity or species level specificity with a broad detection capability. And more recently, we are also using high throughput sequencing as a generic test. MALDI-TOF, we don't use that frequently, but that is also an option as a generic test available. But what is most important for us is, since we'll be looking at nearly 8 different crops and looking for almost 40 different viruses. We mainly on having a test which is more robust, sensitive, and cost effective. We like to have this assays as a kind of a standard that can be used for a long time rather than changing frequently, because we have a strict procedures with regard to establishing standard operating procedures, and changing these protocols can be quite daunting. So we stick with if it is working well, and we generally resist changing it. So I'm not going to providing details of every diagnostics that is available. I only focus on the tests that we have been using. The primary one, the most like a workhorse in our lab, is PCR-based assays, and we use both conventional PCR and real-time PCR, and for RNA for RT-PCR-based detection. And these are being used in all contexts. And for rapid detection, especially off-site or in the field, we have both LAMP, James has talked about it briefly, and we also use another test called recombinant polymerase amplification, which is also an isothermal assay, but that can be used almost at near room temperature. And we find this assay to be more robust compared to LAMP. LAMP is highly sensitive and also prone for contamination, and it requires a slightly elevated temperature for performing assays, which means again the device is required if you have to use in the field. So therefore we switch to recombinant polymerase amplification assay. And we use this assay quite often coupled with ICT apps for rapid reporting from the field. So in case if you find something, a new or even existing viruses, we can readily report, and these apps can provide real-time maps. We have seen the examples both from Wilmer's presentation as well as James' presentation. Less frequently, but also use high throughput sequencing based diagnostics. This is mainly for germplasm health testing for international distribution. And this is a new addition since about 3 years we have been using it. Since emergence of new viruses, especially this outbreak in East Africa, there is a heightened awareness among the regulators who are now demanding more thorough testing before germplasm is accepted. So what we are doing is, as a kind of a general rule, now we are screening all our stocks with high throughput sequencing and establishing a certified seed stock as to use as a nucleus material for ready distribution, ready propagation and distribution. In terms of virus detection, I already mentioned our focus always is on having a test that works very well across all contexts. We have seen the mention of the cassava multiplex PCR in Wilmer's presentation. We have established a multiplex PCR for detection of both cassava mosaic, all the cassava viruses that are distributed in Africa using 3 generic primers. This was published in 2008 by Alabi. It was a joint work between IIT and University of Washington, funded by USAID. And we found this primer set to be highly robust, and they distinguish ACMV and complex of EACMVs. There are more than 10 species, and these primers detect all of them. And we also found that these primers would also pick Southern African cassava mosaic virus, which is localized in Southern Africa. But we have not done wide validation because the sample occurrence of the SCMV is much low in the areas of our interest. But these primers have been shown to be very robust, and we have been using it since more than 10 years. And even when we make modifications to our assays, for example, when we develop RPA, but still target the same region, so that we already know that the sequences that are aligning this, these primers are highly conserved, and we already know that they work very well, so that we don't have to do a validation. And on top of that, we also included cassava brown streak generic primers to it. So in a single assay now, we can pick all the viruses that are present in Africa reliably. We frequently check these primers again as the NCBI database to make sure that the new sequences that are being added can also be detected by these primers. Or if there is any variant, then either we come up with a new primer set or we modify the existing primers. But thankfully, so far, this primer seems to be performing very well. And another unexpected finding as a consequence of the work that was done in Southeast Asia by Wilmer and his team was that these primers that were established in Africa were also known to detect Sri Lankan cassava mosaic. So this is kind of a interesting finding, and then we further try to validate this with collaboration with CTCRI, whether these primers can pick even ICMV and then other variants of Sri Lankan cassava mosaic virus that are available in Makesh Kumar's lab. The data that has shared by Makesh indicated that these primers are very robust in detecting Sri Lankan cassava mosaic virus. But when it comes to Indian cassava mosaic virus, there seems to be, it is not detecting all variants. So we still have to work some changes, and we identified there are certain nucleotides, especially at the 3' annealing site, which are variable. And now we are trying to insert universal codes to see if that would improve the robustness, so that these 3 primers can be used as a universal diagnostics for detection and diagnosis of all the cassava mosaic viruses. So more recently, we are now focusing on isothermal assay, which is recombinant polymerase amplification. As I mentioned, we initially had a LAMP, but we now shifted to RPA-based assays because of convenience. We basically translated the same primers into RPA chemistry. The beauty of this system is that we can readily detect viruses without extracting nucleic acid. And also these kits are quite easy to manipulate and use in the field, because the reagents come as a ready-to-use material. All we have to do is add water and the primers. Although the recommendation is to use, these come as a ready package in a piece tubes, with the recommendation to use 50 microliter reaction. But we found that even at 11.5 microliter volume, they worked very well. So each kit now, although it is supposed to be worked for 100 assays, we use it for 400 assays. That way we dramatically reduce the cost. We mainly use this for detecting in the field or for quick detection for when we are testing material for seed certification purposes. So these are it comes in a kind of 2 formats. The basic format, which we can test like in a normal PCR, perform RPA and then separate them in the gels, or exo RPA, which has a probe with fluorophores that can be used for detection using a real-time PCR or any detector with fluorophore detection system. What we realized was that the sensitivity and specificity of the RPA is similar to that of PCR. It almost has exactly same level of sensitivity, whereas LAMP offers much more sensitivity compared to PCR, but RPA sensitivity seems to be on par with the conventional PCR, which is still good enough for us, because it reduces the contamination risk. And we have established this XORPA, as I mentioned, for real-time detection, which requires a fluorophore detector, which we have now a Jenny-based mobile system, which is battery operated, which can also be taken to the field. This RPA system has been now formatted for multiplex. We are still, it has a little bit issues with the background when we are using in a multiplex format for detecting both ESCM and ACM under the same reaction. So we are now trying to adjust and change the probes to see if, with the different fluorophores, to see if we can prevent that background noise. And the system is also very reliable for detecting virus in whitefly, single whiteflies. And also we have the same system now standardized for cassava brown streak using the same primers. Okay. So one thing that we do is to integrate all of this into kind of field-based system coupled with a mobile reporting app. So these assays can be performed readily in the field. It takes about 15 to 20 minutes with no DNA extraction step. It makes even more convenient for detection. And in case if a virus is detected, then it can be reported directly from the field. So this is something that now we are promoting broadly for surveillance of not only cassava viruses, but also for other viruses of quarantine importance. The last diagnostic that I would be introducing to you is high throughput based virus detection. We construct small RNA sequencing libraries and use them for sequencing using next-gen sequencer, and then analyze the data using de novo genome construction methods. This is a protocol which we established in-house in collaboration with Jan Cruz of SIP as part of a BMJ-funded project recently. So now we have full capability for in-house library preparation and a bioinformatics pipeline. We use two different software, a virus detect, which was established as part of the project, commercial software, openware software space. Both offer strengths, and we realized a combination of both actually offers much better output. This is just to share. I won't go into too many details in the interest of time, but last year we we have been using it since 3 years. In the first 2 years, we have been outsourcing library construction, but since last year, we have performing everything in-house. We constructed 41 libraries. We often test a single sample or multiple samples. We are getting fairly good reads. What is most important is the correlation between the high throughput sequencing based detection and conventional PCR or RT-PCR. So of 41 libraries that were analyzed, we have observed correlation of 90% between PCR and high throughput sequencing based assay. Whereas in case of 2 samples, we got positive detection in high throughput sequencing, negative detection in PCR. In another 2 samples, we have a positive detection in PCR, but negative detection in high throughput sequencing. The library quality was good, and we still don't know what contributing to this this differentiation. It probably is not surprising in case if a sample is positive in high throughput sequencing because relatively high sensitivity, but negative in PCR. But what we don't understand is the negative results in high throughput sequencing. And we have tested this thoroughly. I don't know, we probably would like to learn from some others' experience, but so far what we realized was this correlation between high throughput sequencing and PCR, which is quite encouraging. And what we detected in these 41 samples, most of these samples actually tested were from West Africa. We detected West and Eastern Africa. We detected the standard virus, cassava bigamo viruses and the brown streak viruses. In addition to that, we have detected cassava ivorian basiliform virus, which is an annular virus of family Bromoviridae. This virus was reported in Cote d'Ivoire first time way back in 1980s. And more recently in 2014, it was sequenced in Scottish Crop Research Institute. They had a cassava sample procured in 1990s, and they have done some high throughput sequencing they published by Stuart Gray and his team. And this sequence has 99% correlation. So this virus detection is less frequent, but otherwise we have not detected either virus so far. So now shifting gears and moving on to the use of diagnostics for clean seed production, which is a very, very important in terms of applied diagnostics. I move quickly from here onwards because I will be talking about well-known procedures. Basically, the procedures we use for generating clean seed is a standard thermotherapy and meristem tissue induction in vitro. And this has been in use for over 50 years at IIT and has been working very well for both cassava as well as cassava mosaic as cassava brown streak elimination. But the only problem is that this approach is quite slow, especially from in vitro bulking to the rapid propagation, which is often necessary for clean seed systems. So in 2016, we have adopted a new method called semi-autotropic hydroponics. This is a propagation system that was adopted for potato propagation in Argentina. So we adopted this system for cassava, for direct propagation of tissue culture plants in SAH system for rapid bulking. So it's a very simple system, basically can be used at normal aseptic conditions under with a basic hygienics that in a open laboratory. We take tissue culture plants and then plant them in a substrate, and the plantlets readily root, and the same can be further cut and rapidly establish planting material in boxes, which can be either transplanted directly into the field or taken into screenhouse for further hardening and for further stem production. So it's a kind of a game changer with regards to increasing the rapid propagation of cassava Now, for example, in a very small, about 10 into 5 square meters, now we can generate almost 200 to 250,000 plantlets. And also this dramatically increases the throughput. We have been using this system now as a standard approach for generating clean stocks. So for tissue stem cuttings on backwards to either in vitro planting material or SAH propagation. See, like in case of in vitro plants, even if you use thermotherapy and meristem, little infection coming through some of this, the clones that were coming out of the meristem. So we have to test and eliminate those so that virus-free planting material for propagation. And now we also have established a new propagation method bypassing the in vitro propagation to generate cassava, virus-free cassava planting using SAH systems. So that is also giving the same level of, the same percentage of clean planting material production. So we use both of this for establishing virus-free nursery, virus-free stock nursery, and the stem cuttings harvested from this are also used for international distribution. So by combining all these methods, now we have established a kind of a pipeline for rapid propagation of cassava, starting from the field to establishing virus-free planting material to propagation in SH, and then bulk production under nurseries or greenhouse, and the field propagation. Now this has been integrated into the conventional seed system, which is mainly based on stem propagation, which involves 3 categories of seed production, like breeder seed, foundation seed, and certified seed, with each generation limited by a certain number of cycles. This is a system that is regulated by the National Agriculture Seed Council of Nigeria. And the same material can also be used for international distribution. And this is where, again, the regulations play a big role, and the policy and the diagnostic tools. There are different systems depending on the country and whether the distribution is for international purposes or domestic distribution. So diagnostics are mainly used for establishing clean stocks as well as health certification, whereas the policy of the national or systems govern the type of procedure used for establishing clean stock and the method used for moving the material. So there are too many procedures operating at the global, regional, and national level, but ultimately the operations happen at the national level. So whatever the country that is binding, whereas the international and regional policies provide a kind of a broad framework. So we have been exchanging materials almost 5 decades, and I'll just give one example of our recent experience of moving material from Africa, East Asia, basically to move CMD resistant varieties from Africa to Asia as a counter to controls SLCMV outbreak in Cambodia, Vietnam. So the material has been shortlisted by the breeders, and they have to be distributed. They have been propagated as per the procedures that I explained earlier, the classic system. And then these were dispatched. But during the dispatch, we have encountered a lot of challenges with regard to getting permits from these countries. All I can say is that this material now have been successfully transferred, and they have now been tested, as you could see from the tweets from SEAT. This is a kind of a good example of a technology established in one country moving to another country to solve a virus disease outbreak. The challenges that we faced is the permits. For example, the procedures for distribution material, exchange of material with Vietnam and Thailand, although they are in the same region, we are sending exactly the same material, is completely different, and it can actually a lot of things, especially with regard to the time it takes to move material. So we have been systematically characterizing and documenting the regulations and the conditions used for importation of material from Nigeria. So we have exported, for example, material to 42 countries in the last 10 years, these almost 201 distribution events. What we noticed was that in about 31 countries, there are no specific guidelines. They simply say, okay, you need to oblige with the standard phytosanitary procedures and send material, so they don't specify any viruses. Whereas 8 countries are providing specific conditions. For example, they specifically want tests to be done for virus A, B, C, and then results to be provided before approval is given. 2 countries have changed in the last 10 years. They changed their regulations. And 1 country has been inconsistent. Sometimes they use specific conditions and sometimes they are not. So we are learning from these experiences, and now we are, regardless of what the countries say, we follow the standard approach, so that there is no risk of any virus spread from the country of origin, and also have all the documentations recorded, because this is very sensitive issue. And in case virus outbreaks happens, the first thing they look at is from where the material has come, whether that material has been a source. So to address these differential regulations, we are now working with the regulatory agencies and the IPPC to establish a kind of a green pass phytosanitary protocol system for distributing pre-certified and pre-tested material from CGR centers. So we have made fairly good progress. The whole material is tested, and this will be accredited by the regulatory agency, and there will not be any further bottlenecks to move this material. So my last topic is on domestic distribution of planting material. So this is where now we move into the seed systems that was mentioned by James also. The thing with cassava is that the cassava seed systems is highly fragmented and dominated by the informal seed producers. So literally we can say that there is no structure. So when we think of formalizing a system with as a kind of pipeline delivering clean seeds, then we need to strengthen the whole value chain. So this is a kind of the existing system dominated by the high, almost 90% dominated by the informal seed producers, about which we know very little. The so-called formal system catering to about the 5% of the market. So since 2016, we have a project called Basics, which is focusing on strengthening the formal cassava seed systems, especially to establish a system which is commercially sustainable and viable and operate on its own without subsidy. So as a consequence of this project, now we have a prototype as strengthening production of breeder seed to foundation seed and certified seed with various multiple agencies engaged here, with the quality assurance from movement of material from the nucleus to down to all the way to the root production. So when there is an involvement of multiple stakeholders, there is always a challenge of fragmentation, and the communication becomes a big. And this is where we have come up with a digital solution called Seed Tracker to integrate all the cassava value chain actors. So this system can be used for any crop, but in this case, we have tailored it for cassava, and I will be giving an example for Nigeria system. And this system has been tailored to the regulations that are existing in Nigeria. So it is a web app. There is a website which is open access. Anybody can review information. And this provides the basic details about the regulations and the best procedures to follow for cassava seed production. And it also provides the details of where the clean seed production is happening, and then the various value chain actors that are distributed in Nigeria. The best thing is that this system has been tailored to the regulatory the procedure for cassava seed production, which involves the three stages of inspection before pre-planting, post-planting, at the time of harvesting. It also digitized all the information about the released cassava varieties so that one can readily verify the variety information for access. We also have established new standards. Previous standards were outdated, and we have revised it. As James mentioned, we have stringent standards for viruses. For example, brown streak is 0 because it doesn't exist in Nigeria, whereas cassava mosaic is an endemic problem. There's little tolerance. 0 is only maintained at nucleus seed and breeder seed. And there are some other endemic fungal and bacterial diseases for which maintaining 0 is not possible. And so therefore, we have a kind of a threshold which is based on the severities. The system is now well adopted and is in use since 2017. And what you see on this is now the clean cassava seed production fields of different types, breeder seed or foundation seed. It is now possible to access all of it through the seed tracker system, and it is also possible to curate information directly from the map and also check variety details, and if interested can make order directly through online form, or also make contacts with WhatsApp, which is now becoming a very popular mode for communications. So since establishment of the system, as you can see, there is a great improvement in the formal seed production of cassava. Each dot that you see is on the map. Each year there are about 100 to 100 cassava seed producers are now being registered as commercial seed producers in Nigeria. And another important aspect is we could also monitor the spread of varieties and the change over a period of time. We can see which variety is now adopted much faster, which variety is replacing over a period of time. All of it is possible because the entire data is available in a single system. More recently, we also introduced an Android version of this to further simplify access of this tool to the users. And now it is possible to register seed fields using this app and also check variety information and also make purchases. We are promoting since beginning this year. Variety information is one of major challenges in the past. Now it is made simplified. It has been simplified. Using this app, it is possible to check details and also verify the field registration possible. The moment field is registered, once it is up to the regulator, it appears on the map, and through the map to access details and make contacts with the producer. The whole advantage of this system is to encourage people to register and also gain market access, because one of the major challenges having information about where to procure quality seed, now that has been made, that has been simplified through this application. It has various other features, for example, product advertisement for both root and stems, again as an incentive to promote products of the users of the seed tracker app, and also option for ready buying using this application. One of the best thing that happened during this development is that the regulatory agency, that is National Agricultural Seed Council, and to find this app to be something very useful and adopted as a national platform, a tool for e-certification, not only for cassava, but also for other crops. So this now has a very simplified system now for registering seed producers through the NAS seed tracker app, through their portal. So this is a kind of a game changer with regard to formalizing the information, especially of the crops which you don't have a very well-established seed systems such as cassava. And this is really helping to foster a robust seed system for vegetatively propagated crops in Nigeria, and also now being piloted by James in Tanzania. We're also now looking at to further scale this into other countries. So as a kind of a summary and a way forward, we have a very complex task at IITA. And so far we have been doing good thanks to availability of sensitive, versatile, and robust diagnostics. We have been successfully moving planting material from IITA programs across almost to 40 countries worldwide, in almost in all continents. We have now standardized a very well established system for high ratio propagation for driving cassava seed systems, which is playing a very big role in strengthening formal cassava seed system, coupled with the digital tool called Seed Tracker, which is also now adopted as a formal platform for e-certification. But having said that, there is always a threat of emerging diseases. We always wary of new diseases coming in. So this is something that we always have to prepare and be ready to establish new tools and also new phytosanitary protocols to eliminate them from our stocks in case if they are detected. So the risk of unknowns remains a big threat, and we remain vigilant for that. With that, I would like to acknowledge some of the team members who have been making a big role for this, the work that I have presented here, and acknowledge our donors who contributed to this work. Thank you very much.